Hello, sophomores. Welcome to your next episode of the Sophomore Diversity Seminar. Dr. Smith and I are here with you today to chat with you about the question, who am I? And I'm going to do my best to convince you that the best answer to this question is, I am my biology. Let's start with the basics. I am my DNA code. I inherit from my parents all those sequences of A's, C's, T's, and G's that are giving the information on making me. I am my genes. That's certainly my biology. I am my amino acid sequence and I am my enzymes. Those genes are coding for the sequences of amino acids that make up my proteins, that make up my enzymes, that control everything that goes on in my cells. So, for example, here's a little bit of a DNA code that's making up hemoglobin molecule, codes for the messenger RNA that codes for the sequence of amino acids making up the hemoglobin protein. If one letter were to change, say this T to an A, that would cause a different amino acid from glutamine to valine to get placed in the hemoglobin protein. And I would no longer have normal hemoglobin. I would have sickle cell hemoglobin. And if I had been born with sickle cell anemia, I would have spent probably many episodes in a hospital. I would have had a number of sickle cell crises during my childhood, and I pretend that I would be a very different person as a result of that one letter difference. So I am my biology. I am my amino acid sequence. I am the union of one egg and one sperm. My father could have produced 8 million different kinds of sperm, 8 million different kinds of eggs from my mother. Yet I am the union of just one of each of those, making me one of 64 trillion different possibilities, all the result of that meiotic process. I am a unique individual because of my biology. I am 23 plus 23. I inherit 23 chromosomes from my dad, 23 chromosomes from my mom, giving me my 46 chromosomes most of the time. Sometimes something happens during the process of meiosis, so that instead of having the normal number of chromosomes, I might get an extra chromosome, like an extra chromosome number 21. This fellow right here happens to have 3 copies of number 21, and he has Down syndrome. I suggest to you that if I had been born with 47 chromosomes and had Down syndrome, I would be a very different person than I am now. I would not have been a biology teacher at John Burroughs School. I probably would not have married the man I did. I probably would not have raised 3 children. And all of that is a result of my having 46 chromosomes or having 45 or 47 chromosomes. I am my biology. I am my sex chromosomes. I inherit 2X chromosomes if I'm a female, an X and a Y chromosome if I'm a male. Although I want to suggest to you that those two possibilities, XX and XY, are really just two ends of a continuum, and that there are a lot of things in between, as you will see in a moment. In order to understand those variations, we need to look at a little more biology. I am whether or not I inherit an SRY gene. The SRY gene is on the long or the short arm of the Y chromosome. It stands for the sex determining region of the Y chromosome gene, and it codes for a transcription factor. It codes for a turning on a particular gene, which starts a cascade in individuals who have the SRY gene. So if I inherit that SRY gene, it's going to influence what gonads I have when I am 6 weeks of age in the uterus. So just barely developing. Gonads begin, but you can't tell whether those gonads are going to develop into testes or into ovaries. But under the influence of that SRY gene, the gonads begin to develop into testes. And if that gene is absent, they develop into ovaries and all of the other plumbing that gets associated with it. Well, I am certainly my biology, depending on whether I get testes or ovaries. And along with that, along with the testes, come certain hormones and receptors. Take a look at this diagram because it's important what we're going to talk about in just a moment. That SRY gene starts a whole cascade of interactions. So the testis makes a hormone called anti-Mullerian hormone that's going to cause any female structures to degenerate. So if you didn't have that SRY gene, you wouldn't have anti-Mullerian hormone and you would develop as a female. On the other hand, when that SRY gene is present, it causes an interaction that goes on inside of the testes in these interstitial cells. It causes cholesterol to get made into testosterone. And that testosterone is going to act via receptors on the cells to be responsible for the internal structures on the male. So the epididymis, the vas deferens, the seminal vesicles, all the inside stuff comes under the influence of testosterone. Then testosterone gets converted to another hormone. It's called DHT for short. Similar to testosterone, but it's a little bit different. And it primarily, again via receptors, is responsible for the external structures. The penis, the scrotal sac, a little bit of the inside, but mostly the external structures. Testosterone for the inside, DHT for the outside, all being mediated through receptors. Now, I could be a variation on this theme of XX and XY. Here's some individuals. They're XY, they have the SRY gene, means they're making testosterone, they're making DHT, but they have no receptors. They don't understand, their bodies don't understand that they have testosterone present. And so, when you look at these individuals, I would say that these individuals look female, yet they are XY and with the SRY gene. We don't have a good vocabulary to describe these individuals. I can be another variation on this theme. XY have testosterone, have receptors, but I miss the enzyme that converts that testosterone to DHT. So I don't have DHT. These individuals, when they're born, mom and dad will look and say, oh, this baby looks like I have a little girl. And then around puberty, something interesting happens. At puberty, the adrenal gland starts pouring out androgens. It's pouring out testosterone-like compounds, or I should really say like DHT compounds, and structures that we had thought of as being female come under the influence of these hormones and begin to develop in the male pattern. So here's an individual at 19 and at 42 years later. Most of us would probably look at these individuals and say male. And if they were standing naked in front of us, you would say, yes, they have the male structures. So here we have an individual that more or less begins on the female side of things and converts to the male side of things. Here's a third variation. These individuals, XX, and have an adrenal gland where the feedback loop is not working in its normal pattern, and it causes them as a fetus to pour out those large numbers of testosterone, large numbers of androgens. And even once they're born, that adrenal gland continues to pour out those androgens. And so, under the influence of all that testosterone, an individual who is XX can look very much like this individual here. And I would probably look at this individual and say, that person looks like a male, and he was raised as a male. So this was probably in the 1940s before we have the medicine that we do today. Today, at birth, we can correct that outpouring from the adrenal gland, and individuals would not have that excess testosterone as an adult. But interestingly, they still had that testosterone as a fetus. And so now we can ask the question, does exposure to testosterone influence my behavior? Is my behavior my biology? And if you look at some studies, the answer is yes. Here is a study that looked at sisters. One sister had the excess testosterone in the fetus. It was corrected at birth. The other sister did not. So very genetically similar, raised in the same household, yet look at their behavior. Very little interest in child care by those exposed to the excess testosterone. Very little interest in pregnancy and motherhood, weddings and marriage, playing with dolls. Not much difference in about careers and marriage. But the other things, very different kinds of behavior. So my biology is my behavior. What about my brain? My brain is certainly my biology. Is that different? Look at male and female brains while just reading and listening. Here on the male side of things, using the right side of the brain. On the female side of things, they tend to use both sides of the brain. I am my brain. I am my biology. What about my sexual orientation? Is that my biology? It can be very difficult to tease out the contributions that the environment and genetics play for something like sexual orientation. What's a cause? What's an effect? Here's a couple of examples, both of which are hypothetical examples. So I could have, for example, a close binding mother, something in my environment, that might induce a hormonal, a biological change that leads to homosexuality. Or I could have facial beauty, something genetic, that might influence my parents to treat me in a certain way, something in my environment, that would lead me towards homosexuality. Telling apart cause and effect is difficult. What biologists do to try to answer that question is to look at identical twins. Identical twins have the same genes, and so if one twin is gay, is the other twin gay? If sexual orientation is due to your genes, if one twin is gay, you'd expect 100% to be due to, let's say you'd expect 100% of the other twin to be gay. Not quite what we find. We find that if one twin is gay, 52% of the time, the other identical twin will be gay. Now, compare that to the normal incidence of homosexuality among the general population. That's around 4%. So 52%, way more than just the random picking two individuals from the population. So yes, my genes make a large contribution to my sexual orientation. What about the other way? What if we take two adopted individuals? So no more related than any other two random people from the population. Are they more likely to be gay if one brother is gay? The answer is yes, there is a slight increase, 11% compared to 4%. So the environment does play a role, but not nearly as much as the genetic factors. So I am my biology. I am a Homo sapien. I am my evolutionary heritage. I inherit things because I'm a primate. I have opposable thumbs. I can do things with those opposable thumbs. I am a primate. I must feed my young with my mammary glands. I must produce my young in my uterus. I need the help of a mate to raise my young because they're born very underdeveloped. So I am the product of my evolutionary heritage. I am my geographic ancestry. Take a look at these three pictures. They're showing genetic variation that exists among the human species 100,000 years ago, 65,000, 40,000 years ago. The colors are representing the variation that's present. So here, 100,000 years ago, lots of variation. We hadn't yet really moved much out of the continent of Africa. We started moving, we started migrating. We brought with it some of that variation. Now, today and 40,000 years ago, we have occupied most of the globe, but all of the variation that was present in Africa didn't make its way out of Africa. If this blue circle represents the variation in Africa, people today in different parts of the world are really just subsets of that variation that was present 100,000 years ago. But am I really very different from people around the world? And the answer is not really. Yes, if you look around the room, you can describe all kinds of things. Do I have an attached earlobe or a free earlobe? Can I roll my tongue? Do I have a widow's peak? Do I have a hitchhiker's thumb? But all these things that I'm mentioning are really very superficial kinds of differences. When you get down to how I'm functioning, to how I'm working, when I look at the pepsin in my stomach, we all have nearly identical pepsin molecules. Look at the hemoglobin molecules that I was talking about earlier. We are very, very similar. Little bit of variation. Most of that is pretty superficial. So I want to leave you with I am my biology. Whether I'm looking at my DNA or my genes or my enzymes, I am my biology. Thank you.